very, very complicated, and there is plenty of specific cases. What I want you to remember about what we saw yesterday is essentially the part of the non-coding sequence compared to the part of the coding sequence, and the importance of what we call the junk DNA, which is the non-coding DNA. And it is quite important in terms of evolution processes. So yesterday we covered most of the aspects of the genome structures, and I just want to finish what we saw yesterday with a few slides about one concept which is very interesting. It's in the field of what we called comparative genomics, which means what kind of information can we get when we compare two genome sequences from different species, and what is underlying this aspect. So we will see today a few slides, I would say in half an hour, something like that, about this comparative genomic aspect, and then we will shift to the second part of the lecture, which is the structure and the regulation of the chromatin compaction. Okay, so I start back. So basically, the slides that you are going to see now, they will be in the same PDF file as the one as yesterday. Okay for everybody? Okay, perfect. So one second, I try to share my screen. Right, it's this. Okay. Do you see the gene duplication and the importance of gene duplication in in adaptation of plant and animals? So the slides that I wanted to show you yesterday is this part. So this is the end of the lecture of yesterday. So it's the comparison of the macro structure of the genome. And this is called what we call centening. Have you ever heard about this word?
No, I personally haven't.
Okay, so so you understand when I write comparative macro structure. So it means we can compare the structure, which means the order of the sequence between different genomes. And the centeny, if you check on the internet the definition of centeny is a notion. So it's very general, and then when we apply that to genetics or comparative genomics, it means that there is the same orders of object between two situations. So concretely, it means when we use the word centeny, we don't say, how can I say, sorry, we can say there is a conserved centeny. If there is a conserved centeny between two genomes, it means that the order of the object of the sequence, so the object of the sequence can be the genes, can be the repetitive sequences, they are in the same order between two genomes. Okay, so this is what we call centeny. We call that the conserved centeny between different genomes. So there are very interesting things, and I want to show you a few ways to access this kind of data. So I won't go inside all the bioinformatical processes to produce this kind of data, but I will show you some kind of very key results that the comparative genomics can give us. So the first of all, so you remember that all the coding sequence and repeated sequence are ordered and they are located in different chromosomes. So wait, I'm going to try to put the pointer. And if we look in depth of the sequence of chromosomes between different related species. So in this example, there is that's the chromosome number 2. So a schematic view of the chromosome number 2 coming from human, and the same data, which means the same coding sequence, so the macro structure of the same loci in the monkey chimpanzee. Okay, what happened is so the monkeys and humans are extremely closely related. There is something like 99% of sequence identity if we compare locally the genomic sequence between human and monkeys. But if we look at the macro structure of the chromosomes, so of the genomes, we can realize that, so you have like dark shade green bars that correspond to different loci in the chromosome. Then you've got light shade. This part here is the centromeric region, okay? And on the north and on the south, you've got the telomeric regions. So in human, the whole content of the gene that are present in the chromosome number 2 in humans, it is part of 2 different chromosomes in the monkey, okay? So I don't have the number of the chromosome there, but what happened during evolution, so you know there is a specific number of chromosomes in human. Chimpanzee, the monkeys, they have a different number of chromosomes, but the content of the genomic DNA is almost the same. So what does it mean in terms of evolution? In terms of evolution, we can see this process in the 2 ways. Either it is a fusion, the chromosome 2 from human comes from a fusion of 2 ancestral chromosomes in monkeys. So this chromosome fused to this one, you lose a centromeric region, this one or the other one, and then from 2 chromosomes, you have only 1 chromosome at the end. But so it means when we want to compare the macro structure of the genome, we can compare the number of chromosomes. So in that example, the chromosome number 2 of human is the result of the fusion of 2 chromosomes from other monkeys. But the order of the loci in all the chromosomes, they are exactly the same. Okay? So have you ever seen this kind of process? The fusion of chromosomes between species?
No.
No, okay. So there are, well, okay. So here, so it's a way to represent this kind of fusion and other genomic rearrangement. So you don't have to learn that completely, but you have to understand. So here, so look at the title. We write down synthetic relationships. It means the relationships between the orders of the gene in mouse compared to human. So there is a color code. So first of all, we're going to see, I'm going to describe you a little bit how this figure is done. So on the left, you've got the 22, so from 1 to 22 down there, the 22 autonomous chromosome in human. Then you've got the X chromosome and the Y chromosome of human. So all the left side of the slide is the structure of the 24 chromosomes that are present in Homo sapiens. On the right, it's exactly the same representation, but when we are talking about mouse. And the mouse has, instead of 22 normal autosome, chromosome, non-sexual chromosome, it has only 19. Sorry, somebody is coming. So the mouse has, 1 second. Okay, I have to. Okay. So the mouse has, instead of 22 in human chromosome, normal chromosome, autosomes, it's called, it has 19. And it's also, the mouse also, because it's a mammal, it contains the sexual chromosome X and Y. So if we, once we have the full genome sequence of the mice genome and the human genome, so it means that we got every single read of the genomic sequence of every chromosome in these both species. We can compare the genomic sequence between the two different species. And so this is where the color code is coming from. So how do you have to read this kind of data? So we're going to start, for example, for the chromosome, all the data, it means all the genomic sequence data contained in chromosome 1 in mouse, they will be spotted in the human genome in red. So it means that when you see a red piece of sequence in one particular chromosome in human, it means that this sequence is coming from the chromosome 1 of mice, chromosome 1 only. And so on. For example, yellow boxes, if we look at here, the top of the chromosome 1 in human, if you look at the color, it's like a kind of yellow range. So it means that the north of the chromosome 1 in human is the same as part of the chromosome 4 in mice. Okay? So do you understand how to read this kind of graphics? Is it okay? Okay. So what you see, if you want to have a global analysis of this, if you check all the different bits of color, so for example, the whole sequence contained in chromosome 1 in mice, you find it back in human, but it's not in one single chromosome. It will be located in different parts of different chromosomes. So you see, for example, chromosome 1 of mice is the sequence contained in chromosome 1 of mice will be present in some parts in chromosome 1 in humans, some parts in chromosome 2. You have it also in chromosome 6, and so on and so on. Okay? And so it's a way to compare where some specific genomic sequence located in one specific chromosome in one species, where it is located on the other chromosomes of another species. And this is what we call centenic relationships. Is it better now to the definition of centeny? Yes or no?
A little bit more clear.
It's a little bit more clear. I'm going to show you another way of representation, because this way of representing the correlation between genomic sequence between two species. So if we look at the chromosome 1 of mice, we see very easily that all the sequence of chromosome 1 is spread in different chromosomes in human. But we don't know where the sequence, for example, on the top of the chromosome 1 of mice, where is it located in the different chromosomes of humans. So there is another way of presenting this kind of data, and it's like that. So don't be afraid at all. You will see that very often. One second, I need to turn off the pointer. 1 second. Sorry, because I have plenty of windows in my computer. Okay, so here it's the best way to represent the centeny relationship between two species. So I explain you. So it's extracted from a paper published in BMC Genomics in very early in 2021, last year, and it's the centeny map of pigs and human genomes. Okay, so we are in a situation. It's mammals. Both of them are mammals. And what they did, so the full genome sequence of pig is available. The one from human is available as well. And they did a very impressive bioinformatic analysis of the sequences of these both genomes. And then they compared. So how to read that? So it's a circle, okay? And in this circle, you've got different, so let's start, for example, here. So on the top, it will be easier. So you have this little gray or different colors, okay? And on the top, you have HS or SC followed by a number. So if you look at HS stands for Homo sapiens. So this is chromosome 1 from Homo sapiens, chromosome 2, chromosome 3, and so on and so on. And if you go down, you see that there is 22 chromosomes for human. So in this map, they don't put the X and Y chromosome. We only stay for the 22 chromosome autonomous in human. So we've got the 22 chromosomes from human, and exactly the same thing coming from pig. So chromosome 1 for pig, 2 for pig, 3, 4, and so on. And you can have the number of chromosomes in pigs, it's 18. Okay? And then there is those are, you maybe won't see it, but it's not important. Here you've got numbers for every chromosome. You've got numbers, which is basically the position in the chromosome in megabase. Okay? So I will take, for example, this example here. We are in the chromosome 3 of human. Here, this is the position 0, and here it's the position, so this is the top of the chromosome, and down there it's the bottom of the chromosome. Okay? Exactly the same for chromosome number 4. The top of the chromosome, so the north, is position 0, and the south is position, I cannot read here. So here we have a clear representation of the sequence from the bottom to the top of the chromosome. And when you do bioinformatic comparative genomics, you can take, for example, the whole sequence of chromosome 3 in human, and you check what are the sequences that are identical in the same order in the genome of the pig. And you realize doing that, so if we take the example of chromosome 3 of human, so you see all this green line, they correspond to match sequence between the chromosome 13 in pig and the chromosome 3 in human. So I don't know if you see it very, very well, but some bits, if we take here, so it's the bottom, you see the 0 is here, so the top of the chromosome 13 in pig, the bottom of the chromosome 13 of pig. And can you see the mouse and this dark green color that is going from 13 going to this 21? Can you see it? It's okay? So what does it mean? It means if we take, for example, this part of the genome in human, so the top of the chromosome 1, it is completely identical to the middle of chromosome 13 in pig. And for example, the bottom of the one bit of the bottom of the chromosome 13 in pig is duplicated in the chromosome, is also present, in the chromosome 21 of human. Okay? So, 1 second, I try to... Okay. So do you understand how to read this kind of map? It is basically the relation because of the identity of the sequence of one part of the chromosome from one species to other part of the chromosome of another species. So here, what can be the conclusion? So can you try to make the conclusion on the comparison between chromosome 3 in human and chromosome 13 in pig? What would you say if we ask you the question, what is the relation between these two chromosomes in terms of genomic structure? Can you answer that?
I think there is a very close syntonic relationship between these two chromosomes.
Very strong. Okay.
A strong relationship between these two chromosomes. However, a very small part of the pig chromosome 13 is, however, lies very, related to the human chromosome 21?
Exactly. So here you can, this is exactly that. So here you can point out one putative chromosome fusion. For example, if we take chromosome 21 in human, you see that the chromosome 21, all the data contained in chromosome 21 is found back in chromosome 13 of pig. And the rest of the sequence coming from the chromosome 13 of pig is found completely in chromosome 3 of human. So basically, we can say, without any big mistake, that chromosome 13 in pig could be a kind of fusion between the chromosome number 3 in human and the 21 in human. Okay, so I hope everybody can see that. There are plenty of other examples if you read carefully. So there are some kind of so the example between chromosome 3 in human, 21 in human, and 13 in pig is quite simple. Okay, because most of the 3 in human is found back in the 13 in pig. But if you look at, for example, the chromosome number 6 in pig, the syntenic relationship between pig and human is very much more complicated, because if you look at the whole sequence of the chromosome 6 in pig, it has been divided during evolution and split between some part of the chromosome 1 in human, the bottom of the chromosome 1. You have back in chromosome 16, you have it also on chromosome 18, you have it also on 19, and even on 20, a bit of in 22. So all those sequences present in a single chromosome in the pig genome, they are also present in the human genome, but in different chromosomes. Okay, that's the way to read this kind of map. And so you have some very strange relation, like the chromosome 6, and then you have a very strong one. One which is very strong, for example, is the chromosome number 5 in pig, is almost 100% of the chromosome number 12 in human, except this little bit here that is going to the 22 in human. Okay? So that's a way to represent the structural relationships that we can find between two genomes coming from two different species. So this kind of map, we use this kind of map when we want to compare the whole genomes. But we can do an analysis of the syntenic relationship at a very smaller range. So this is an example, and we will almost finish with this. So if you look carefully this drawing, so I just explain you where it comes from. So it comes from a website which contains all the genomic and genetic data from C. elegans. So this is the model worm that we saw earlier. So it's a kind of repository of all the genomic sequence of this species. And if you look, I will put the pointer 1 second. So in this website, it's a public website. You can enter different loci, and you have the comparison of the genomic sequence of different Caenorhabditis species. So here we compare 3. The model 1, you see here, is called reference, is C. elegans. C. elegans is a model organism. It's completely sequenced. We know almost everything about this little worm. And there are the genomic sequence of a very close relative to C. elegans, which is called C. bridgei, and another one which is called C. renai. Okay, so here we are dealing with 3 different species of the same genera, Caenorhabditis, and we are looking at some portion of different chromosomes. So, for example, the blue part on the top, so this is the genomic sequence of C. bridgei. So you see here the rule it's written in kilobase. So we are really zooming on a very specific loci in the genome. So here, this is the same portion of the genome, but for C. elegans, and in red, it's the same portion of the genome from C. renai. So 3 species, extremely closely related, 3 genomic sequence. On the top of this box here, you've got this little drawing. So what do you think it can be? Do you have an idea when you see something like that? Or like this at the bottom, and here as well.
Is this the structure of the
what?
gene of the genome? We have the repeat sequence, and we have the exon. The square exon or something.
So the square, they are the exons. So this bit, for example, it's a gene.
Yeah.
Okay. So this is a gene. This is a gene. Here, there is a gene. Another one, another one, another one, and another one. Okay. In all the gene models, so when I say gene, it's the coding part. So the little boxes, they are the exons, and the lines there correspond to the intronic sequences. Okay. So here we've got a gene that is going to be transcribed. I don't know if you see there is a small arrow there, which is on the right for all the gene model in this line. And the little arrow is on the left for gene model in this one. Do you see it? So there is a different color. So this is purple, 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 and the arrow is on the right. And for the blue one, the arrow is on the left. Do you see it? Yes or no?
Yes.
Okay. So it's important because I will explain you something. So it means that I'm going to take this example first, and we're going to compare with the other results. So it means that in this individual, in this piece of DNA, we've got 1, 2, 3, 4, 5, 6, 7 genes. Okay. There are, among these 7 genes, we have 1, 2, 3, 4, and 5, the purple one, that are transcribed from left to right. Okay, so it means on one strand of DNA. Then there is 2 other genes in this region, which are transcribed from right to left. So it means on the other strand of DNA. Is it okay for you, this? Tell me, yeah, because
Yes, it's okay.
because what you have to keep in mind is that in genomic DNA, this is double-stranded DNA, we can have genes that are transcribed from one way and from one strand, and other genes that are transcribed in the reverse way from the other strand. Okay, so we can have different open reading frame like that. So what's happening? This region contains a certain number of genes in either one orientation or the other one. Okay, that's perfect. Then if we compare the nucleotide sequence of this C. brigée, so this species, if we compare to C. elegans, here we have in blue what is exactly the same and the orientation. So if you look at carefully, there's a little bit of strange things. We're not going to talk about this one. If you look at, it means that this position here in the blue one will be exactly the same as this position in the C. elegans genome sequence. And this one is the same as this one. So we found, what do we find? In brigée, there are 2 genes, but in elegans, there are only 1. And if you look at the site of this bit of the sequence, when we compare these 2 species, you see that this one in brigée is bigger than in elegans. And we found back this open reading frame, this gene, is exactly the same as this one here. So it means that this part of the sequence in this species is exactly the same and has the same position in the other species. But this gene has been deleted because you didn't find it back. Okay? But the order, the global order is the same. Then if we move, and we found, if we want to go on a little bit further, we have this part of the genome, which is very similar in terms of sequence and orientation to this part in elegans, and the same for Raynaud, exactly the same, and we found back the gene. So there is a strict correlation between elegans and Raynaud sequence. You see only one open reading frame, same orientation, and a very, very conserved microcentennial there. When we compare elegans to Breguet, it's a little bit more complex. We've got a very good conserved centennial because the order is the same, but there is a small deletion between these two species. Okay? So that's an example. And I just want to come back in this last example here, in the middle of the drawing. Here you see that there are crosses like that. And this is very interesting. What it means that the genome sequence of this individual, Breguet, in this region, so from position 95, let's say, to 80, okay, something like that, it contains two genes. If you look at the orientation of the open reading frame, the genes, they are from left to right. Okay. So the first one is a small gene. The second one is bigger than the first one, but both of them are in the same orientation. If we look at the same loci, same genomic sequence in the context of the global genomic sequence in C. elegans, we found back these two genes. But the small one is on the right and the big one is now on the left. And the orientation of the open reading frames of both genes is reversed compared to this one. So what does it mean? It means that the global information that is contained in these regions between these two species, the information is the same, but the structure of the information is a little bit different. So the beginning on the left part, there is a very, very conserved synteny because there is collinearity between the sequence of the three species. Even there is a small deletion. But if we look a little bit more in detail, this centered region, we see that this piece of genomic sequence in Breguet is also present in C. elegans, but it has been reversed in the genome. So it means that the information stored in this piece of DNA is still present, is just reversed. And we write it like that. It means that this position in Breguet corresponds to this position in elegans, and this position in elegans corresponds to this position in Breguet. And exactly the same concept there. So it means that this sequence in C. elegans that we found reversed compared to Breguet is also found reversed in Raynaud. Do you understand the idea?
Yes.
You can tell me. So if we want, I just turn off this. One second. So if we compare the previous slide, this one, where it's a global at the chromosome level comparison between different chromosomes in different species, you see that can you see the mouse here? Okay, so you see that, for example, we see that when we use this kind of map, we see only a gross correlation. So we know that this part of the chromosome is found back in another way, but we don't know exactly the orientation. And when we look more closely to the comparison of the sequence, we found to this level of detail. So we found collinearity in some situation and inverse collinearity in other places. Okay, so when you see this kind of analysis, this kind of data, 3 species, the genomic sequence analysis of the 3 genomic region, and this kind of correlation. What you can see, and you can draw an hypothesis on that. We have among the 3 species that we study here, we've got 2 of the 3. So the first one, Brigae, and the last one, Renae, that are exactly the same. So the order between the red and the blue is the same. And what we can see here is that it is C. elegans that contain the inversion, but not the other one. The other one, because they're supposed to be the ancestral genomic structure of this species. And what can happen is that in C. elegans, there is a bit of this DNA that has been reversed during evolution, but the content of this fragment is still the same. You understand? Okay, so why do I explain you all these kind of things? Because it's a very good point in order to do genome assembly. I will explain to you a little bit. If we start, so don't remember this, but I just want to show you one use of the synteny, the concept of synteny between species. We will see that at the end of next week. It's when we do some kind of physical mapping of the chromosome. So here there is a strategy. We won't go into the detail. I just want to highlight 2 points. The idea in this strategy was, how can we use the genomic sequence of a species, which is Arabidopsis thaliana, that has been completely sequenced. So first thing that we have is the full genome of one plant, Arabidopsis thaliana. This kind of strategy can be applied to animals, to insects, whatever. So you've got a reference genome, fully sequenced. So that's the case of Arabidopsis thaliana. Then what we wanted to do is to have the structural composition of the genome of a very close relative to Arabidopsis thaliana, which is Arabidopsis alleri. Is the species that I talked to you yesterday that was able to grow on highly contaminated soils. Okay, so we have the sequence of thaliana. We don't have the sequence of alleri, but how to take advantage of the conserved synteny that exists between these 2 species. That's the idea in order to reconstruct the map of Arabidopsis alleri. So the idea, and we will finish with this, I just show you. So you don't have to remember that, you have to remember the strategy. The strategy was very, very straightforward. So we will focus on the top of this drawing. So the top line here on the bottom correspond to the genomic DNA sequence of Arabidopsis thaliana. Okay, here the writing on the top, they correspond to different genes in Arabidopsis thaliana. So there is a way to give the names of the gene. So it's always the same for all species. So AT means Arabidopsis thaliana, 2 is the chromosome number, G is because it's genomic, and this is the gene number. So you see that this part of the genome in Arabidopsis thaliana correspond to the chromosome number 2, and you have the genes. Okay, so, and the order is like that. Okay, gene number, tak. So it means that in this, it's 1 megabase region. we've got something like 10 or 15 genes in Arabidopsis thaliana. What has been done? So you see that in Arabidopsis thaliana, the order of the gene is known, is well known. We have the full sequence. All the bottom part of the drawing correspond to genomic fragments coming from Arabidopsis alleri, not thaliana. Thaliana is on the top. Alleri, this is the different fragment here. So it's a kind of library that contains very big DNA fragments from the other species alleri. And what we can find is, in order to reconstruct the map of alleri, so the map would be this clone, then this one, then this one, then this one, and so on. So you can have the full genomic sequence. Don't worry, we'll come back on that later on. But if we check for the presence of specific gene from Arabidopsis thaliana in alleri, we can check the presence of every individual gene in different genomic fragment from alleri. And what we can see in this drawing is that it is completely conserved. What I want to say is, for example, this one, the gene 130, it's present in this fragment. In thaliana, the 230 is just by the 130. And what happened is we found it back in the same genomic fragment, and so on. The 230 that is present in this genomic fragment is also containing the next gene in thaliana, in alleri, and so on and so on. So you can see that when we can take advantage of the conserved synteny between different species in order to reconstruct the genome sequence. What I want to say saying that is that since the order of the gene is mostly to be the same between these two species, we can use this order to order different genomic fragments once compared to the other, and then reconstruct the whole genomic sequence. Okay? Is it clear, this aspect? Can you tell me? So the advantage of this conserved synteny, it's quite important because it can be very useful when we, so one example would be, okay, we have a species that is already fully sequenced. So we have plenty of genomic data available, but you are interested in a very closely related species, but you don't have any genomic data for this new species. But what can be done is to use the conserved synteny between these two closely related species in order to take advantage of the same order of the genes in order to find back the structure of the unknown genome. So it can be very useful because it's going to speed up very much the time needed for sequencing a new species. Because once you have the order of the genes in the new species, it's really faster to do the global genomic sequence. Okay? But we will see that later on. Okay? So do you have, so this is what I wanted to say to you yesterday, but I didn't have time. So it's just to, so you won't be, I won't ask you a question about the conserved synteny. The idea is that you have in mind that there is some common feature between genome, between different species, and the fact that the order can be conserved between closely related species, it's a great advantage in order to have new genomic sequence of unknown species. It's really easier. Okay. Do you have any question for now?
No, I don't have any questions.
It's okay for everybody?
I have one question.
Yes, please. Oops. I cannot hear you. Can you try the microphone? Okay. I saw the message. Sorry about that. Wait. Your mic is not working. Okay. But okay, you can ask the question when it will be okay. Okay, so now we can shift to the second part of the oh wait, it doesn't work. Sorry. Okay, I have to share. Do you see the slide now? Okay, because I've got a small screen. Okay, so we're going to go a little bit. Okay, so all what we saw before, so yesterday and just the beginning of today, is the global structure of the genome. So important to know, but not completely necessary to understand in terms of gene regulation. So now we're going to go a little bit deeper in the chromatin structure and essentially the dynamic of the chromatin structure. So it will be a little bit maybe complicated for you today, but so this is important that you understand that now. So I'm going to explain you or to show you how chromatin is structured, what it is. And you will see there will be plenty of information about the dynamic of the condensation of chromatin. We will see that in a completely different way, starting from the day after tomorrow on Thursday, because I will explain you how the dynamic of the structure of chromatin is extremely important in terms of gene regulation. And we're going to talk about gene regulation and the central dogma of molecular biology. And we will talk back again. So today, what I want to show you is the basic and main feature of chromatin. And then we will use this concept later on in the lecture in order to understand how chromatin structure can influence gene expression. Okay? This is what we call the epigenetics. So the chromatin structure and the dynamics. So we will start by very simple things that you know, but at least you can come back on that. The DNA compaction is completely compulsory because in every eukaryotic cells, because we have to fit. For example, the human diploid genome represents something like 6.2 or 0.3 gigabase. Okay, it means that the size of the DNA, if you take the molecule of DNA, I mean the 22 molecule of DNA contained the 22 chromosome, if you tear this molecule, in human it represents 2 meters. So in one cell, in a single cell of your body, your DNA represents 2 meters of DNA sequence. And you have to fit this 2 meters of DNA sequence inside the nucleus, which is 6 micrometers. So you need to compact everything. Is it okay for that? So that's really important. So the chromatin, what it is? The chromatin, the definition of chromatin, it is the full genomic DNA plus the protein. Okay, and what kind of protein do we find very tightly associated to DNA? There are histones, so that you know this protein, and we are going to talk a lot about histones. They are the major proteins that we found in chromatin. We have also other proteins that are specifically associated with genomic DNA, which are transcription factor. We will talk about all the transcription factor Thursday and Friday. We have general transcription factor. We've got specific transcription factors. Then we have all other protein components, so RNA and DNA polymerase, and plenty of other factors. So chromatin refers to genomic DNA plus different proteins that are always associated with DNA. So this is the 5 level of compaction or organization of the chromatin inside the nuclei. So you know this, it's just to remind you. So we can take it from the bottom or from the top, it's the same. So at the bottom we've got the representation of a chromosome that corresponds to a chromosome during the anaphase of mitosis, because it's compact chromosome, we draw it like that. And there is the 5 level of organization. So we can start from the top. We can go very quickly on that. So this is naked DNA. You know that DNA is structured around the nucleosome. We're going to talk a lot about that. So this is protein complexities that surround DNA. Then there is a second higher order of compaction. Even so, this is a condensed molecule, then you have this, and then it turns to be the chromosome. So you see that from a very, very long, that's 2-meter molecule of DNA, you can have a chromosome because of this compaction. So that's the first point. When we look at the DNA contained in nuclei, so this is an electron microscopy of a nucleus at the interphase. It's very important because the different phases of mitosis, we will have different type of pictures. So during the interphase, we have most of the cells in our body are either in interphase or G2 phase. The nuclei look like this. So this is, we have the nucleole. Do you remember what is the nucleole in the nuclei? Do you remember?
Is it where the RNA is?
Okay. Yeah, this is the site of synthesis of transfer RNA and ribosomal RNA. So it's a little bit darker because of the electron. But what we found is 2 types of chromatin. So it's the words, they are coming from very long time ago. So we have lightly colored chromatin and dark color chromatin. So we call that euchromatin for the light color and heterochromatin for the dark color. Okay. So there are 2 kinds of chromatins. Then we're going to have a look at what's happening during different phases of mitosis. So during G1, S, and G2 phase, this is most of our cells in our bodies or in every living bodies are in this stage. So the DNA is not really condensed. This is a representation of DNA that is compacted. In green, this is the matrix of the nuclei, and all the genomic DNA is linked to the nuclei envelope by some protein that we call condensin. That's not a problem. But during the M phase, so during mitosis, where there is separation of the chromosome, what we have is that this structure chromatin is going to be highly condensed in order to form the individual chromosome that will be separated during the late telophase. So from that, we have 2 different state of chromatin. And we're going to talk, for now, we're going to talk about these two states of chromatin. So here on the top, you've got two electron microscopy of DNA with protein. So this is very high resolution microscopic pictures. And we can find DNA in let's say, two different states. Either something like it's beds, different beds spaced with DNA, and other where these beds are extremely condensed and stuck together. So the schematic of these two states is just there down there. So we have part of the chromatin that we called euchromatin, which is not condensed. It contains nucleosome, but nucleosome are separated by what we call here, I will show you a picture later on, linker DNA. And on the other hand, the other state of chromatin is highly condensed. So you see there is a super order of organization. So it's a spiral of this relaxed state. And we call that heterochromatin. So in that state, the DNA, which is in blue here, is extremely packed around the nucleosome. We're going to see that. And all of them are in interaction. So we've got a very highly compacted stage that we call heterochromatin and a relaxed state that we call euchromatin. And what I want to talk to you about today is what is the relation between the compact chromatin, so heterochromatin, and the relaxed chromatin, euchromatin. And we will see that there is a relation between the state of compaction and the gene transcription. And we will talk about that for the next lectures. Okay, so what is heterochromatin? Heterochromatin, extremely dense packed. So this is a very similar picture. Here we see very clearly some heterochromatic region, and here as well. So that's electron microscopy of a nuclei as well. So heterochromatin, the key point to remember, extremely packed. And heterochromatin state can be comparable to the structure of the chromosome during mitosis. You know, during mitosis, you've got the two arms of the chromosome that are extremely packed, and then they're going to be separated in the two daughter cells. At that stage during mitosis, the genetic material, so the DNA, is extremely packed. If we compare the state of heterochromatin to this chromosome during mitosis, it's extremely similar. So this region of the genomic DNA are extremely packed, and the gray one, they are relaxed. So one key feature of heterochromatin, and this is what is interesting us, is that heterochromatin is mostly transcriptionally inactive. So what does it mean? It means that in a general manner, it's not all the time. It's always like that. It's not 100% the case. But a region of the genomic DNA that is in the state of heterochromatin, which means compact around the nucleosome, if there are genes in this region, they cannot be transcribed. Why they cannot be transcribed, according to you? Do you have an idea?
Because the DNA is too compact.
Mm-hmm.
It is twisted and bound together, and I think in that way then the
bone cannot connect to the DNA.
Okay,
and so it cannot start the transcription.
Exactly. So you are saying exactly the same thing. So the compaction, the result of the compaction, in order to have transcription of one gene, you need to have all the promoter sequence that is accessible to the RNA polymerase II, transcription factor, and so on. But when the DNA is completely packed in heterochromatin, these sites of interaction with RNA polymerase II and the transcription factor, they cannot find the sites. So it's written, it's mostly transcriptionally inactive. It means that in almost all the case, all the machinery of transcription cannot reach the DNA target. So you cannot have transcription. But it's written just down there because the chromatin, you can have access to the template for RNA transcription. And one point also, because maybe you didn't know that, but during mitosis, you don't have any transcription at all because every single chromosome is so compact, you don't have any transcription. So it means that in the late stage of mitosis, when the chromosomes are packed and are separated in two daughter cells, all the mechanism inside the cell is coming from already existing proteins. There is no transcription, okay? And then transcription starts back again when the two daughter cells are separated, okay? So there is a first point that is interesting us is that the nature, the state of compaction of chromatin is going to have a direct link to the possibility of transcription of specific locus. And this is going to be important in terms of gene regulation. For heterochromatin, it's a little bit more complex than euchromatin. So I didn't talk to you about euchromatin, but you may find that it's very easy because it's exactly the opposite way of heterochromatin. So before talking about what is written in this slide, I just tell you that heterochromatin, because it's compacted, you don't have any transcription possible. At least most of the transcription is not possible. But as a comparison, euchromatin, so the relaxed chromatin, you have active transcription. So it's a little bit black and white. Heterochromatin, no transcription. Euchromatin, transcription possible. Okay? But in heterochromatin, there is one specific point that is very interesting, is that we've got two kinds of heterochromatin. When I say two kinds, they are the same, biochemically, or almost the same, but they are different in terms of behavior. So we have something that we call constitutive heterochromatin. So the characteristic of this constitutive heterochromatin is that it's very resistant to decondensation in interphase. What does it mean? It means that during the cell cycle, during mitosis, so if we start from G1, we've got one single copy of the genetic material. During S phase, there is duplication of the genetic material. G2 phase, we've got two copies of the genetic material. Then all the stage for mitosis, so anaphase, telophase, metaphase, and so on, you have compaction of the chromosomes, then separation of the chromosome into two daughter cells, and then the cycle starts again. But what happened is after mitosis, when the new daughter cells enter in G1, transcription need to start again in order to have the cell that can live, okay? But what happened for constitutive heterochromatin is that these regions are compact during G1, S, and G2, during mitosis as well, but stay compacted in the next generation G1, S, and G2. So this is what I write when I say it's relatively resistant to decondensation. It means that this constitutive heterochromatin exists during interphase. Is it okay? Yes or no? Tell me. Okay. Then, on the opposite, we've got this facultative heterochromatin. What makes the heterochromatin facultative, and this is where it's the most interesting, is some regions are found in the heterochromatic state in some cells, but not in other cells. So it means that in one body, depending on the organ, you can have some cells that have some specific locus that are completely condensed, heterochromatin, and other cells, just by the first one, they can be decondensed in this locus. So it's completely reversible, and what we found in this facultative heterochromatin, it does not contain a lot of repeated DNA. And what I forgot to tell you just a little bit before is that the constitutive heterochromatin, we found it is essentially associated with repeated DNA sequence. And obviously, we found it by the centromeric region and the telomeric region, which are composed by repeats. So what is the conclusion of that? We've got the two kinds of heterochromatin. Constitutive, it stays compacted in every state of the cell cycle. And the localization of this constitutive heterochromatin is mainly on repeated DNA sequence or centromeric or telomeric sequence. According to you, from what we saw yesterday, what can be the rationale of having repeated DNA sequence in a compact form of chromatin? Do you have an idea? We already talked about that a little bit yesterday.
Can you repeat the question?
So, the constitutive heterochromatin, the characteristic is that it will be condensed in all the different stage of the cell cycle. So during the interphase, during the mitosis. And if we found back this constitutive heterochromatin mainly in all the region of the genome that contain repeated sequence. Okay. So from what we saw yesterday, you remember that I was telling you that, for example, in human, we've got almost 50% of the human genome that is composed by repeated sequence. This repeated sequence, they can be either transposon, retrotransposon, line, sign, and so on. So if you remember yesterday, I was telling you that retrotransposon or transposon can jump from one place to another place. The fact that a transposon can jump from one place to another place, it's very interesting in terms of dynamic of the genomes, but it can be very dangerous because it can produce mutation. Okay, you remember that?
Yes, so if it's in the constitutive, so if we keep the transposon silent and it
exactly.
Okay.
You're completely right. So it means that this constitutive chromatin is here to protect part of the genome that cannot be stable. It means that all this part of the genome that is composed by transposon that can jump from one place to another place, they need to be shut off. And they need to be shut off during all the cell cycle, whatever the stage. So this constitutive heterochromatin is mainly produced in order to stabilize this repeated part of the genome that are highly unstable. Okay? And because this part of the genomes, they are completely compact, transposon cannot jump. Not all the time. Sometimes one transposon jump, but most of the transposon cannot jump when they are in heterochromatin context. Okay? If we compare that to facultative heterochromatin, and for us, in terms of gene regulation, it's much more interesting facultative heterochromatin than constitutive one. Because facultative chromatin means something very important. It is dynamic, which means that some region in some cells can be relaxed euchromatin, and the same region of the genome in a different organ or different cell can be compact. So it means that according to the state of compaction of this region in different cells, some part of the genome can be expressed in some cases, and in other cases, they are not expressed. Do you understand what I want to explain? We will see that in a few minutes. Okay, so for this facultative heterochromatin, it's going to be something that is extremely finely tuned in order to control gene expression. But we will make a synthesis of that. So we're going to talk about the regulation now. Do you have a question? Yeah. I was going to say, do you have a question? Tell me.
So because the heterochromatin is tightly condensed like that, can it be replicated during the interphase, like especially in the S phase? Because yes. I don't know if it can, the DNA pole can bind to the DNA or something like that.
Mm-hmm. Now, that's a very good question because it depends the way we present it. So you are completely right. At one point, DNA should be completely relaxed in order to be duplicated. And it cannot be duplicated. This is what you are asking. It cannot be duplicated if it condensed. Okay? But look what I wrote here. The constitutive, it's relatively resistant to decondensation in the interphase. This is, when I write this sentence, it's not totally 100% condensed during interphase. And we are going to see that when chromatin is condensed in a specific locus, there are some molecular actors that can locally decompact chromatin in order to do either replication or transcription. But your question is very interesting because in theory, if this constitutive heterochromatin was always completely packed, you cannot have a replication. You're right. But during the S phase, you will see that there are some local point of decompaction that allows the replication. Is it okay what I explained?
Yes, thank you.
And to be sincere, and it's very important what you say, because but it's very complicated. So let's take, who spoke in French? Sorry, I've got something strange that is happening to my computer. Somebody. Aha. Sorry, 1 second. Okay. Now to come back to this. The question you ask is very, very interesting and complicated. So, constitutive, can you hear me? Is it okay? Because I turn off, yeah? Okay.
Yes.
So constitutive chromatin always compact. Always compact because the most important things to do for constitutive heterochromatin is to avoid transposon jumping from one place to another place. Okay, so that's the key point of heterochromatin. And I will explain you something after, because there are clear relation between constitutive heterochromatin and cancer as well. So during the interphase, you are completely right. The genetic material needs to be duplicated in order to have two daughter cells. So it means that during the interphase, the cell has to decompact the constitutive heterochromatin. It's compulsory in order to make the replication. But it has to be very, very short in time and very localized. So what is happening is inside the cell during the S phase, you've got local point of constitutive heterochromatin that are decompact. Then this decompaction allows replication, and as soon as the replication has stopped, the chromatin is compact back again. Okay, so it's very, very quick. But during this very small time of replication of constitutive heterochromatin, what can happen according to you? You can have transposons that are jumping. Okay, so what I was saying to you, it's heterochromatin. This is what is written on the slide. It's mainly localized in repetitive DNA, so it means in sites of genomic DNA where you have plenty of transposons. And this is to ensure that the transposon won't jump from one place to another place. But on the other hand, during replication, you need to decompact a little bit to do the replication. And it's during this replication time, or meiosis, it depends, that you can have a few transposons that can jump. And then, as soon as replication or meiosis is finished, all this constitutive heterochromatin is compact back. So it means that there is a kind of window of time during replication where the heterochromatin is naked, and it's at this time that the transposon can jump from one place to another place. Is it okay? Do you understand?
Yes.
So you have decompaction of constitutive heterochromatin, but in a very short time during and extremely localized during replication. It's compulsory, but it is dangerous for the cell. Also dangerous because you can have transposon jumping from one place to another place. Okay? Is it okay, or do you have other questions? Okay, we go on. So this is a drawing of a nice drawing of chromatin, and we're going to study a little bit the biochemical component, the protein that are structuring all these chromatins. So in this drawing, you have the two states. On the right is the condensed one, and on the left it's the relaxed one. So euchromatin here and heterochromatin. So you see the difference of compaction. So what I want to show you here, it's only to show you the proteic partner. So we have in purple here what we call the nucleosome, the core nucleosome. It's composed by it's an octameric complex, so 8 monomers of histones. We're going to see that in details. And then there is another histone, which is called H1, which is not part of the core nucleosome, but which is linking the two rounds of DNA, because there are 2 rounds of DNA around the core nucleosome. So this is the way and we will talk about this H1 histone, because histone H1 is going to, and it plays a very important role. It's not only H1, but it participates a lot to the shift between relaxed chromatin and compacted chromatin. I don't know if you can see in this drawing, but when we shift from non-condensed to condensed, what is happening is that you have the nucleosomes that are going to be very close together, and it's because of interaction between H1 and other proteins. So you see here, for example, the H1, they are closely related, what makes the compaction. So we'll come back on that. What I propose you is that we make a small break, because we are going to study all the different proteic component of the nucleosome. I'm going to talk to you about post-translational modification of histones and the relation between post-translational modification of histone and chromatin compaction. So what I can propose you, we make, what time is it? It's 25. We make a 15-minute break, and then we finish the lecture after that. Is it okay for you?
Yep, it's okay.
Okay, so it's 25, let's say at 40. We meet back at 40. Okay. Okay, perfect. Thank you. So see you in 15 minutes. Thank you. Okay, everybody is back again. Okay, apparently yes.
Yes.
Okay, so we go on a little bit. So I put in full screen. Okay, so as I was telling you, we are going to see a little bit what's happening in terms of protein biochemistry on this histone protein that are composing the nucleosome and that are directly responsible for chromatin compensation. So this is the on the right. You've got the first paper. So I didn't put the date. See, it's 97. In 97, there was the first crystal structure of the nucleosome at a very high resolution, 2.8 angstrom, that has been published in Science. It was in Science. It's a very important publication. So in the right part of the slide, you see how DNA. So DNA is just around here with the double helix that you can see. And in the middle, you see that the main alpha helices of the histones. So there is a color code. So we cannot see it really well in this picture, but there are 8 monomers of histone, and there are different histone, H1, H2, H3, and we're going to see a little bit how it works. So the crystal structure was quite complicated to have because it's not a crystal structure of only protein. It's a combination of DNA and protein that has been crystallized. This is why it was complicated. So we see clearly that, so it's 1/2 of the nucleosome. We see 1 round of DNA, and here we can see at least 4 histones. And if we look at on the other side of the nucleosome, we can see that there are 2 rounds of DNA, and in total there are 8 molecules of histone. So basically, on the left, you've got the schematic strategy that was used to understand how was structured the histone in the nucleosome. So it's a quite straightforward strategy. So the researcher, what they did, they took DNA. So we can extract DNA without removing the proteins that are directly linked to DNA. So here, this is the typical configuration of euchromatin. So it means relaxed chromatin. We've got the nucleosome, 2 rounds of DNA around this octameric complex, then a linker DNA, which is a part of the genomic DNA that is not involved in the nucleosome, and the core histone, which is symbolized by this orange cylinder. So how to access the composition and the size? There are 2 slides for this aspect. So what they did, it's very simple. Anyway, in molecular biology, we don't have a lot of method. The idea was, let's take a piece of DNA with the protein that are already in interaction with DNA. If we digest this DNA with a nuclease, which is non-specific, it can cut every bit of DNA without any sequence requirements. So what is going to happen, it's written here in the drawing. So you've got random cuts everywhere in the DNA, everywhere where the nuclease can have access. It means that the nuclease can cut only in the piece of DNA where there is no histone core, because the presence of the protein, and you see here on the right, the histone octamer completely protect the DNA from being cut by the nuclease. So the strategy was this one. We use a nuclease that is going to cut only in regions that are not protected by the octamer. At the end of that, you can collect only the complexes with DNA and the protein composing the nucleosome. And after, because this type of interaction is weak interaction, they are ionic interaction. So if we increase a little bit the concentration of salt, we can separate the DNA content from the protein content. This is what is written here. And then after, it's classical biochemistry analysis, which means from the dissociation of DNA and protein, we can analyze the DNA by itself. And the finding was that the DNA that is rounded around the nucleosome represent 146 precisely base pair of double helix DNA. Okay, so one nucleosome use 146 base pair of DNA to have the two round of DNA. And for the protein part of the complex, you can use different biochemical techniques, such as plenty of different techniques of separation. And the idea was to analyze the composition of monomeric compound in this histone core. And what has been found is that it's composed by 8 different monomers of histone, but different type of histone, because there are several types of histone. So there are 2 molecules of H2A, which is one specific histone, 2 other molecules of H2B, which is similar to H2A, but with a little bit difference in terms of protein sequence, then H3 and H4. So another way of representing the structure is this one, and here you've got the stoichiometric ratio of all the components. So typical nucleosome structure, in blue-gray the DNA. Here we've got the red balls. This is the octamer. So you see there are 4 and 4 on the top and the bottom. So what's happening in terms of stoichiometry is that we have one set of a tetramer, which is composed by 1 molecule of H2A, 1 molecule of H2B, 1 molecule of H3, and 1 of H4. So this is a tetramer. And this tetramer is duplicated in 2 copies to make the octamer. So the octamer at the end contains 2 protein H2A, 2 H2B, 2 H3, and 2 H4. So this is the 8 histone, what we call the core nucleodome. So you see that this histone H2 and H3, they are very different. They are similar between themselves, but very different from the H1 histone. And this H1 histone has a completely different role. It's very well schematized here. It's going to clamp the DNA that is warped around the core nucleosome. And you see it stabilized the 2 rounds of DNA. So the total nucleosome will be, it will be 9 protein, 8 histone forming the core of the nucleosome, and 1 H1 that is going to clamp the DNA in order to stabilize this complex. Okay, so that's the structure. We need to know a little bit the structure to understand the function after. Is it okay? Another point also I forgot to tell you is that histone, in terms of biochemistry of protein, they are small protein. Okay, that's not a problem, the size, but they contain a huge amount of amino acids which are basic, like lysine, arginine, and it makes sense because these amino acids, which are very basic, in the context of the nucleus, they can directly interact with DNA because DNA is charged negatively and will interact very easily with the protein that are very basic. That's just a biochemical point. Okay, so this is a little bit more detail about the analysis of the structure of the nucleosome. So it's just for your your information, we don't really know, you don't really need to remember how it was done. It was just to show you that it's very, very old that these nucleosomes are studied, but with very basic strategies. So it was using nuclease in 74, I was only 3 years old, or using detergent. And it has been done like that. So the core particle, this is what I showed you before, host 146 base pair fragment. So there are different levels, and they show that using a very old technique. So using the nuclease, they cut in between the nucleosome, and you see the rest of DNA in this gel electrophoresis. But this was just this slide, sorry, is just for your information on how they managed to understand what was the size of DNA involved in the nucleosome formation. So this is the definitive structure of the nucleosome with the 8 monomer, the 2 rounds of DNA, and the H1 as a linker. What is interesting in this H1, because we talked only about the core of the nucleosome, so it's sorry, there is a mistake. It's DNA. It binds DNA at the entry and the exit of the nucleosome. So you see interaction between H1 and DNA. It's not done in the 2 rounds of DNA, but it's just when the spacer DNA start to enter in the 2 loops and when it exits the 2 loops. There is something that has been found that H1 is responsible to modulate transcription activity. So it's an indirect, completely an indirect evidence, but what has been found, and I was already showing you this in this slide, H1 here in yellow, it's not the only actor of this, but it participates. It will be implicated in the transition from relaxed chromatin. So relaxed chromatin, I remember, it is transcriptionally active chromatin because the gene can be transcribed. And this H1 participates to the condensation with protein-protein interaction, to the condensation of the chromatin. So it makes sense that they found that H1 in one way is responsible for modulating the transcriptional activity, because what is going to happen is, according to the state of H1, it can either participate to the compaction of the chromatin or to the relaxation of the chromatin. And because the state of compaction is directly linked to the transcriptional activity, there is a link between the way H1 works regarding to the transcriptional activity. And this protein interacts with plenty of other factors, and you can imagine that it plays a role in replication and transcription. So H1, what you have to remember, if we talk about this, all these different histones, is that all the ones that are composing the core of the nucleosome are very similar to each other and interact in between each other, whereas H1 is a very different histone, but participate to the nucleosome formation as well. Okay, so this is very descriptive, but we are going to start a little bit of the... so this slide is a schematic view of the assembly of the nucleosome. So it's not very complicated, but it's a sequential phenomenon. So you see, and we will come back on that, there are dimers, which are heterodimers, that can be formed between 2 different types of histones. For example, here we've got a dimer of H3, H4 that can be formed. DNA, and try to keep in mind that histones, they are extremely well structured, but they've got a tail in the NH2 terminal sequence that is outside of the nucleosome. You see, can you see here in yellow? So yellow represents the 8 monomers of histone, H2A, H2B, H3, and H4, the core of the protein. And the little yellow lines that you see here, this is the NH2 terminal sequences of the histone. And if you check, there are 8, because there are 8 monomers there. So the sequential assembly is very simple. You've got monomers that turns to be dimers. DNA can bind to this dimer. Then another dimer can be recruited to form the complete nucleosome, and so on. So first tetrameric particle, and then the final nucleosome. And you see that all the tails, but you have to see that in 3 dimensions. So you've got all the NH2 tails of the histone that are coming out from the nucleosome. And this is going to be very important in terms of gene regulation. So it's just for your information. So this is the assembly process of the nucleosome. Then at the end, you've got the final nucleosome. And all this part of the slide, this is what we saw already before with the structure of the euchromatin here and heterochromatin there, and then a little bit more complex. If you want to have more details on this paper from Morales in 2001, it's a very good synthesis of what's happening for the histone and the nucleosome. It's very well written. Okay, do you have any question? It's okay. So I put this other slide. It's exactly the same as this one, but it's another way to see it. So maybe it will help you to understand more. Exactly the same process. So this is the nucleosome assembly with dimeric formation of H3S4 before and H2B and A after, and then the final complex with the DNA which is surrounded. So both of these slides, this one and this one, they contain the same data. It's just another view of the system. Okay. So one particularity of the histone, I'll just show you the two coming slides, just for your information. So I think that you may understand that because of their function, the histones, they need to be structured as an octameric protein complex, and they bound to DNA. So during evolution, I will show you 2 phylogenetic relationships between different histones. This means that this protein, they play such an important role that they didn't evolve for very, very, very long time. And so these proteins are extremely conserved. And what is very strange, so here you've got a phylogenetic relationship between different species. So we're starting from yeast. So if you look at all these species, they are all eukaryotes, okay? Unicellular for yeast, human, mammals, plant, insect, and always yeast, plant, insect, and human, and mammal. And then there is other things. Here you've got the relationship between H2, H1, H2A, H3, and H4. And you see that all the 5, because we include H1 as well, all the 5 histones are extremely closely related together, whatever the species is. It means that the relationship between the H2B between yeast and human is extremely close. The same for H1, H2. So those proteins are extremely conserved among all eukaryotes. And as an example, H4 and H3. H4, the analysis of the amino acid changes revealed that they are almost 98% identical, okay, from cows to pig, okay, so very, very strongly conserved. And the suggestion from this number is that there were only 1% of change in terms of sequence over 600 million years of evolution. Okay? Technically, in the full sequence, protein sequence of H4, in 600 million years, we've got they found only 2 changes in amino acid. One valine to isoleucine and one lysine to arginine. In terms of biochemistry, valine and isoleucine are neutral amino acid, and they have exactly the same function. And lysine and arginine are basic amino acid, and they carry almost the same function. So it means that the only changes that we can observe over evolution are what we call blind mutation, because they don't change at all the function. So it's extremely, extremely well conserved. And the next one is the same phylogenetic tree, but just to show you, and maybe this is going to shock you a little bit. So on the previous slide, I will put the laser. So on the previous slide, we just look here only eukaryote, because H4, H3, H2A, H2B, and H1 exist only in eukaryotic cells. You don't have this protein in prokaryotic cells. Why? Because what is the characteristic? I mean, the main difference between a eukaryotic cell and a prokaryotic cell in terms of DNA. What is the main difference? Do you remember? You don't remember the differences? Do the prokaryotes have a nuclei?
No.
No. Okay. So they're supposed not to have the need to have compacted DNA. And do the prokaryotes have chromosomes, individual chromosomes?
No.
No. And what is the main characteristic of the genomic DNA in prokaryotes? It's a circular model.
Circular double-strand DNA.
Exactly, circular double-stranded DNA, no nuclei, and so there is no really homologous of histones in prokaryote. But what has been found is that, and if you see this phylogenetic tree, is that in prokaryotes, we don't have histone. Okay, that's clear. We don't have proper histone because there is no nuclei, there is no proper isolated chromosome. But we found that there are proteins, so they call that HPY, it's histone-like or something like that, that are very conserved, because if you check the name, so we've got Archaebacteria, we've got Thermococcus, Pyrococcus, so different types of bacteria. They have proteins that are not histone, okay, not proper histone, but they are histone-like protein, and they have a little bit the same function as in eukaryote. They are responsible because even if the genomic DNA in prokaryote is circular, it can be very big. It has to be a little bit compact. Okay, so you have this histone-like protein that exists in the prokaryote, but they are not at all regulated like the one from eukaryotes. But there is something that looks similar in prokaryotics. Okay, so this was just to show you these things. So we are going to show you now. We're going to talk a little bit more in depth about this histone. We're going to see different post-translational modification of these histones and the relation between compaction and post-translational modification of histones. So we researchers, so this is going to be, it's not complicated at all, but researchers for several years, they propose, and there are every day more and more evidence on that, that there is what they call the histone code. So histone, we have to start, we're going to, so if you look at the drawing there, we are talking about the 4 histones that are that constitute the core of the nucleosome. So I remember that a nucleosome is 8 molecules of histone. So it's 2 molecules of 2A, 2 molecules of 2B, 2 H3, and 2 H4. Okay, this makes the core of the nucleosome. Every histone in this drawing, it's designed like that. So you've got the ball, for example, let's take H3. This part, which is drawn like a ball, is the part of the protein, which is the biggest one, that is inside the nucleosome. And here, this bit, where you see the amino acid sequence, this is the N-terminal part of the protein. Okay? So this is what we call the NH2 tail of the histone. And this tail, so we've got a specific N-terminal sequence for H3, a specific N-terminal sequence for H4, the same for H2A, and the same for H2B. Okay? The little numbers at the bottom of the sequence, it is the position in the amino acid sequence. So, for example, this arginine is a second amino acid. This lysine is the 4th one, the 9th one, and so on and so on. Okay. So what has been discovered is that we're going to talk essentially about H3 and H4, okay, because after it's going to be quite complicated. The researcher, they realized that these NH2 tails of the histone, they are subjected to a lot of post-translational modification. So for the post-translational modification of protein, there are a huge amount of different modification. You can have methylation, acetylation, phosphorylation, ubiquitination, nitroxylation, and so on and so on. There are plenty of different modifications. And if we look, every type of modification can happen on one specific amino acid. So here there is a kind of map of what are the possible post-translational modification of the different NH2 tails of the histones. So, for example, we're going to talk about this one for the lysine of histone H3, that is at position 9, this one here. It can be the site for 2 different types of post-translational modification. So the little purple spot means that this amino acid, the lysine, can be acetylated, but also, because of the red spot, it can also be methylated. So one important point, lysine, an amino acid, lysine in this case, cannot be acetylated and methylated at the same time. So it means that this lysine, for example, can be either not modified at all, or acetylated, or methylated. So 3 possibilities, okay? And you have plenty of different sites where you can have different post-translational modification. And this is what they call the code of histone. Why they call that the code of histone? Because according to, for a specific histone, according to what are the post-translational modification that happen on the NH2 tail, the consequence in terms of function of the histone will be completely different. Okay? So, there are 2 things. We will come back on that just after. If there is a code of histone that exist. Okay, so you have to listen to me carefully for that because it's a little bit conceptual. So let's take an example. We are talking about histone H3, for example. So I show you back H3, this one. This part of the tail, of the NH terminal tail, can be subjected to different post-translational mutation. Okay. According to what kind of post-transcriptional modification, the function will be different. But what does it mean? It means that if we want to have the methylation of this lysine, it means that there are some proteins, some enzymes that will be able to add a methyl group to the lysine. So it means that we have other proteins that are responsible for this post-transcriptional modification. For example, let's take the amino acid number 10. In H3, it's always a serine residue. Serine residue, serine are amino acid with SH, so it contains a sulfur atom, and this can be phosphorylated. What kind of enzyme do you know that are able to transfer a phosphate group to another molecule? What are the name of this kind of enzyme?
Phosphatase?
No, phosphatase remove.
A phosphate relase? I don't know.
It's a kinase.
Kinase.
Okay. So, for example, no, it's very good that you know that. It means that this serine, for example, will be phosphorylated by a kinase. And if we want to remove the phosphate here, it will be a phosphatase. Okay? So we don't need to learn that. Okay. But what I want you to understand that is this NH2 tail, they have to be modified by specific enzyme, and the result of this modification can be read by other process. I explain myself. So I hope you agree that there is plenty of different combinatories. So PTM means post-translational modification. Okay. So we can have plenty of different post-transcriptional modification in the tails of the histone. They can be established by writers. What I mean, for example, if a phosphatase is coming to phosphorylate H3 tail, it's going to add a phosphate on the serine, for example. So this enzyme will write a code because it will put a phosphate on the serine. On the other hand, once the serine has been phosphorylated, in order to have a different function, it has to be read. So there are readers that can read the post-transcriptional modification that already occurred in the NH2 tail of the histone. Do you understand what I mean? It means that we have proteins that can change the modification of the tails and other proteins that can read these changes. This is why they introduced the concept of a code, because a code has to be written, and once the code is written by a specific enzyme, it can be read back by different protein complex. Okay? So we're going to talk a little bit about that. This is very conceptual. So we have protein that modify the histone tail and protein that will respond to the modification of the histone tails. Then what was observed, so we come back on that. And this post-transcriptional modification, as we just talked about, they are reversible. If we take back the example of phosphorylation, you can have a kinase that is going to add a phosphate on the protein, and you've got the phosphatase that is going to remove. So this is a reversible process, and it is very interesting. This. So I show you as an example the case of histone H3 on this. Excuse me. Wait, I just try to put the pointer. Okay. So, how to read this this drawing? Okay, on the left we've got 3 of the 4 different histones. We're going to look at only one. Okay, just to show you how it is drawn. So we've got H2B here, H3 in the middle, and H4 at the bottom. This little triangle here, they call that the histone fold domain. Technically, it's the part of the protein, which is the biggest part of the protein, that is inside the nucleosome. Okay, it's like a part of a pie. So you need to have 8 of those to make the nucleosome. And this part of the protein, this is the N-terminal part of the protein. Then here, the representation are the different amino acids. We already see that. Lysine, serine, the position number in the protein sequence, and on the top, the post-translational modification that can happen. So we already saw that. For example, the lysine in position 9 can be either acetylated or methylated. The serine at position 10 can be phosphorylated. Lysine at 14 only acetylated, and so on and so on and so on. Okay? And this, what they call the code of histone, is according to what are the modification on the tail, what is the function. So we're going to look at only these few lines, and we'll pay attention to this part. So here you have the same N-terminal part of H3. You see this is H3 with different state of post-transcriptional modification. For example, the top one, you don't have any modification. Okay, every amino acid is exactly normal. On the table on the right, you've got what is the modification state and what does it mean in terms of biology. So we're going to start with the second line. Second line, if we have an histone H3 that is acetylated in the position number 14, it's a lysine. Okay, so if the lysine and only the lysine at position 14 is acetylated, okay, this is the type of modification. Technically, in terms of biology, it means that we have gene expression. So what does it mean according to you? Can you tell me what you think about that?
It's the decondensation, right? Of the protein.
Exactly. It means that when the position 14 is acetylated, the consequence of that is decompaction of chromatin, so gene expression. Okay? And you have all the other possibilities. So to be sincere, this is not all the possibilities because there are plenty. And there is one interesting case. If we look at the lysine at position 9, for example, this lysine at position 9 in the situation of H3, it can be either acetylated or methylated. But, so we can see here, these two lines, either methylated or acetylated. Okay, we see here, acetylated, methylated. But look at the meaning of these two different post-translational modifications. When the H3 carries an acetyl group, the consequence is histone deposition. It doesn't mean compaction. You have histones that are forming the nucleosomes. Okay? But when the same amino acid, so it's a single amino acid, when it is methylated here, the consequence is completely compaction of chromatin and no gene expression, what we call gene silencing. So you understand that with only a few modifications of the post-transitional N-terminal tail of the histone, you can completely modulate gene expression at a specific locus. Okay? Do you see that, all of you? Is it okay?
Yes.
Okay. So we cannot make the complete list of all the different modifications and all the consequences. What you have to keep in mind, that this is the main point of what we are talking this morning, is we have the nucleosome. The nucleosome, it can be either in a compact form, nucleosome close together, or relaxed form. And the change between the relaxed euchromatin to the condensed, which is heterochromatin, only comes from post-translational modification of the histone tails. And you have an example. After, there are, no, we're not going to talk about that. Okay. Let's just move about this one. So let's have a look on this slide. So this is an example of what is happening during acetylation of histone, because I want to talk to you about the enzymes that are responsible for the transfer of this acetyl group to the lysine of the histone. So the concept is very simple. We've got the two states of chromatin at the bottom, the condensed chromatin, non-permissive to transcription, and the relaxed chromatin, which is permissive to transcription. So heterochromatin, euchromatin. Okay. You see here what is in the drawing. This is the N-terminal tails of histone H3. And you see there is small red balls, which means that there is a methyl group that has been linked to the lysine. So this is the amino acid lysine with the NH3 plus residue, which make it completely basic. And we've got two different enzymes that works in both directions. So this is different enzyme. There is histone acetyltransferase. What this enzyme simply take a lysine, a specific lysine, and we're going to link an acetyl group to this lysine. So NH2COCH3. Okay. The reverse process is done through another enzyme, which catalyzes exactly the opposite reaction, which is the deacetylation of the lysine. So what kind of conclusion can you draw from that? It means that I can shift from condensed to relaxed or relaxed to condensed only with the activity, relative activity between these two enzymes. For example, if in one particular context there is only histone acetyltransferase that are active, histone acetyltransferase, we call that HAT. If they are active and the histone deacetylate are not active, what's going to happen is that the lysine are going to be acetylated and we shift from condensed to relaxed. On the opposite way, if we are in a situation where the chromatin is relaxed and we want to condense it locally, you only need to remove the acetyl group of the lysine, and then the consequence will be no acetyl group, then condensation. Okay. So if you think a little bit about that, imagine that in this part of the genome, there is a gene. In the situation where this gene is in the context of condensed chromatin, it cannot be expressed. But in order to express it, you just need to change the configuration of chromatin from condensed to relaxed through the activity of histone acetyltransferase, and then the gene will be located in the part of the genome where it's relaxed, and so the transcription can happen. So it means that there are some situations where the activation of a gene only comes from the change of the chromatin compaction. Do you understand this? Or do you have any question on that? It's okay? So it means that activation of the gene, the canonical concept is to activate a gene, you need to have specific transcription factor so it can start. But it's not only this. It is true, completely true, but it's not only this. To activate a gene, you need also to have the gene that is accessible. This is what we talked about before, accessible to the RNA polymerase II and the transcription factor. And this is governed by the compaction state of the chromatin. And the compaction state of the chromatin is regulated by different enzyme activities such as histone acetyltransferase. Okay, so it's an important point in terms of gene regulation as well. We're going to see that in details in the next lecture. Okay, so that's an example. We are in a situation where chromatin is condensed. We want to locally relax the chromatin. The only thing to do is to transfer an acetyl group and it will turn to be relaxed. After, I have to talk to you a little bit about remodeling of the chromatin. And just to show you something that we will see in detail a little bit later on. Okay, so in order to explain you, I will shift to the, try to make you a drawing. It will be easier. Okay. 1 second. Okay. Do you see widescreen? Yes. Okay. Thank you. So there is. I will show you two situations. The first one and the second one. Okay. The first one. So we imagine that there is a part of the genome. Okay. In this part of the genome, in red, it is a gene. Okay. And all this part of the genome, okay, it is condensed. Okay. You tell me if you don't agree. The consequence of this is that the RNA pol 2 plus the transcription factor, they cannot activate the gene, okay, because they cannot access, so no transcription. Okay, is it okay this? So one way to activate the red gene is to shift from condensed to non-condensed. Well, I'm going to okay. So if we want to shift from condensed to non-condensed, we can have the histone acetyltransferase that are going to transfer an acetyl to the lysine, and the activity of histone acetyltransferase, this one, are going to shift from condensed state to non-condensed state. So in the non-condensed state, the gene now is accessible to all the transcription factor, and therefore you have transcription. Is it okay? This. Okay. So that's a kind of situation. But what is dangerous there? When we shift from the condensed locus to the non-condensed locus, it is true that the gene, it couldn't be activated before, and now it is possible, no problem. But the problem is the sequence around the gene. If, for example, they contain transposon, it's an example completely fictive. When we shift from condensed to non-condensed, the transposon, now it can jump. Okay? And this is really bad. Okay? Do you understand what I mean? It means that we are in a situation where there is an important gene that is in a compacted chromatin, and the cell wants to activate this gene. One possibility is to change the post-transcriptional modification of the histone, so the chromatin at this locus is going to be relaxed, and the gene can be expressed. But if there is some mobile element like transposon around the gene, when the locus is going to be relaxed, the transposon will jump. And this is very dangerous for the cell. So there is another way to regulate specifically the decompaction. The idea is this one. We are in a situation of heterochromatin. Okay, so this is heterochromatin. The gene of interest is here. Okay, that's the gene. But all around, it is extremely dangerous to decompact this loci because they contain a lot of transposon. So we want to decompact only this part. Okay, you understand? Without affecting the other part. Do you agree? Do you follow me? It's a way to protect. So from that, we have this system, and this system is extremely important. So this is an example of the first characterized complexes playing this kind of role. They are called SNF complexes. We don't going to talk a little bit about that. So imagine here, this is the piece of DNA, this box. It's a piece of DNA, this box. And here, under the nucleosome, there is the data box of the gene. So this is the promoter and this is the coding region. The data box is the part of the promoter where the RNA polymerase II is going to bind and to start transcription. So if we are in a situation where the data box or the loci is completely compacted or protected by a nucleosome, you understand that the RNA polymerase II cannot bind to DNA. So it's impossible to have transcription. But what's happening is that, so impossibility of transcription, but we want to activate this gene. So there are some, what we call, and this is a problem, sorry it's in French, but there are some situations of some transcription factor, they do not at all activate transcription, they just change the post-transcriptional modification of histone. So what's happening is that we have histone acetyltransferase, okay, that are going to transfer an acetyl group only on the nucleosomes that are in interaction with the 3-SNF complex. When the nucleosome is in interaction with the 3-SNF complex, the histone acetylase, this specific one, can acetylate the histone of this nucleosome, and the consequence of that is that the nucleosome is moved a little bit away from the region of origin. So when the nucleosome is sliding a little bit, it will reveal the data box, and then all the transcription factor and the RNA pol 2 can bind and start transcription. Do you understand the process? Or no?
Can you repeat the process?
The process, the general process is the situation at the beginning is that the compaction of chromatin does not allow the gene to be expressed. But we want to decompact only the gene, not the sequence around the gene. So you have these complexes, which are called 3SNF complexes, that can specifically bind to the nucleosome that we need to move, and not the other one, and that will recruit histone acetyltransferase that will move the nucleosome. In other words, it's like it is like if you are in a situation where here, so this is a piece of DNA, this is a place where we have the gene of interest, a gene, you need to activate this gene. All the part of the genome surrounding the gene, surrounding the gene has to be in heterochromatin, because there are transposons everywhere there, and it's dangerous. So what is going to happen is that the nucleosomes that are surrounding the gene, they can be specifically targeted by the 3SNF. Okay. And when they are targeted to the 3SNF, these nucleosomes, they are going to move a little bit in order to free the gene, in order to be able to be transcribed. So the consequence of that at the end is you keep on the left and on the right, you keep heterochromatic state, so closed chromatin, but at the loci of the gene, you can have access for RNA pol 2. Okay, so it means that there it's relaxed, but here it is still compact. You understand?
Okay.
So what I mean, the difference between global acetylation is quite easy to understand because you globally relax the chromatin. Okay? So it means that if all these histones are acetylated, they will all turn to be euchromatin. So the gene of interest will be transcribed, but around there may be some danger because of transposon. So there is another way for the cell to activate only the gene. Is not to globally decompact chromatin, but locally only on the gene of interest to decompact the chromatin in order to activate the gene transcription. You understand? It's extremely complicated, but
can I ask a question about the swines at LF? Yeah.
Yes.
So like you said that it's going to slide a bit. So how can we control how it slide? How long it slide, or how long it how it can stop?
Okay. I will show you tomorrow, but I don't have it here. So it's a big complex that unfold the DNA, which is making the two rounds. It unfold it. So the core the nucleosome is a little bit more free, and then make a shift. So it's step by step. The sliding is not very long. It's 50 or 100 base pair, but it just slides enough in order to free the data box. It's not a global sliding of all the nucleosomes. It's just local sliding.
But if it just slide enough for the data box to be reviewed, right? But the data box is the size for the factor of transcription. If only that, then if other part is still condensed, how can the gene be transcribed? If the data box only reviews the data box.
I understand, and you're completely right. So in this example, the target is a data box, okay, but there are a promoter can be very long in terms of sequence. So you have the data box is the place where the RNA with the TF2D and TBP is data box binding protein. This TBP and TF2D, this is the protein that are going to interact with RNA pol 2. Okay, so this is only for recruitment of RNA pol 2. You can have exactly the same process as this one, but upstream in the promoter to have a cis element regulatory sequence to be also not hidden. So in this example, I just show you what happened for the data box, but it happens also for other important parts of the promoter that are responsible for the activity of the gene. I'm sorry, but this example is too local, too much local. You have to consider this example in the whole part of the gene, so promoter and beginning of coding sequence. You see what I mean?
Okay, I understand. Yes.
And so there are, I can find you the data. There are a lot of disease such as cancer, some cancer, they are coming from excessive sleeping of the nucleosome. It means that some of the genes, what is a cancer? A cancer is anarchist cell divisions. So it means that this is normal genes, okay, that start to activate cell multiplication and so on and so on. The process is complicated, and I'm not specialist at all. But a lot of cancer are coming from the reactivation of cell cycle gene that should be silenced in heterochromatin during the adult stage. Do you see what I mean? So if there is a local decondensation of chromatin at the wrong place, it can reactivate a gene that has to be turned off. Otherwise, you can have some genetic disease or things like that. Now, what you have to remember concretely is that we have, with this way, the slide that is on the screen now, we can change the compaction with just post-translational modification, but this would affect global changes. They exist and they are important, but there is another mechanism which is a little bit more fine, which is the same concept, which is decompaction, but that are very, very localized in specific loci of the genome, in order not to reactivate the genes that are surrounding them. Okay. So this was an example of this 3SNF complex. I don't know why I put that here. So just to show you here, this is the first one that has been identified. Just to show you that it is a very big multi-protein complex that are doing this kind of thing. You don't need to remember about that. Okay. Okay, so that's a kind of summary for the histone acetylation. So this is another way to see the things. So here in this drawing, you've got the core of the nucleosome. This is the H3. This is H4, the N-terminal end. You see the different amino acids that can have post-translational modification for H4 on the right and H3 on the left. And you see that what is going, I show you this slide only for this. What is going to, the consequence of a post-translational modification of the histone is at the base of the recruitment of this remodeling complex. So what does it mean? It means that locally you've got condensed chromatin. Okay, the loci you are studying is completely compacted. If you have enzymes that are able to modify the post-translational modification of histone H3 at position 23, putting an acetyl group. Okay, so there is an enzyme that specifically is going to put an acetyl group there. The consequence of this modification will be that the 3SNF complex can read this kind of modification. This is why I was talking to you before about the writers and the readers. The writer protein are the protein that are writing the mark. So the writing of an acetyl is achieved by an acetyltransferase. Okay, so these are the writer of the marks. And then the reader are the complexes that are going to be able to find this modification and that will go for a consequence. So in that case, the writer of this mark is an histone acetyltransferase that is going to put an acetyl group in the lysine at position 23. Okay, that's the writer. And the consequence of this writing, this epigenetic mark, is going to be read by the reader. And in that case, the reader is the 3SNF. Because 3SNF is able to do his job only when he finds this kind of mark. You see the difference between the writer and the reader. The writer are the partner that are going to write the different post-translational modification, and the reader, they are the complexes that have an activity that are able to recognize these different marks. You understand the difference between the writers and the readers? It's okay? And we need both, because you need to have some objects that are going to write the marks, and according to the marks that have been written, some other complexes are able to read the marks. And this is why the scientists, they call that the code of histone, because it's a code that is written and it's a code that is read. Okay? So that's an example just to summarize everything. So as a general context for histone acetylation, we can summarize it like that. You've got active chromatin, repressed chromatin. This is achieved only through acetylation and desacetylation. We already saw that before. This is the acetylation complex is carried by histone acetyltransferase complexes, and the desacetylation by histone desacetylation complexes. And what can be interesting is that, according to you, can you tell me what kind of definition would you give for a transcription factor? Because it's very complex, this.
You mean like, is it a writer or a reader? Or?
No. In general, when you have lecture and when you study, for example, the transcription done by RNA polymerase II, we're going to talk about that the day after tomorrow. So the RNA polymerase II is the polymerase, but it needs to be activated by transcription factor in order to be active. And you already studied that before during your graduation. You have what we called in general transcription factor. They are proteins that recognize DNA in the promoter. And once they have recognized DNA, they can directly activate RNA polymerase II. Is it okay for you that? I'm going to make you just a drawing. Sorry, I prefer that we spend time on that because it's okay. Okay, we're going to see that. The general context is this. A gene, you write it like that. Let's say we've got a 2 exon gene, that's E1 and E2. We've got the transcription start, written by +1, so this will be the messenger. Here we've got the data box. Okay, and here we've got what we call cis regulatory element. Okay, data box. What's happening in the data box? Data box, you've got the whole complex of Pol2. Is it okay, this? But in that situation, if there is only the RNA polymerase II, the transcription doesn't start. A transcription factor, a lot of people are used to call transcription factor a protein that is able to interact specifically with a sequence of DNA. And after this interaction, this protein is able to activate directly the RNA polymerase II, so transcription starts. Okay? You know that? Yes or no?
Yes.
Okay.
Yes.
But what I'm going to talk to you, it's a different situation. Let's go back to the slide. It's similar but different. And the problem is that we call that the same name. Look at in this one. This is why I put it in this slide. Here we've got the transcription. Here we've got the transcription start, okay? Where there is the TXN, it's supposed to be the RNA Pol2 and so on. But look what's happening here. Here in green, you've got a transcription factor. This transcription factor recognizes DNA. So this is exactly what I drew before. But instead of directly interacting with the RNA polymerase II to activate it, it doesn't do that, this transcription factor. The transcription factor recognizes a specific sequence of DNA, but instead of interacting with Pol2, it interacts with histone acetyltransferase complexes. And the consequence of that is that the presence of this transcription factor brings at this specific loci the histone acetyltransferase. And in that case, it will put some acetyl group in the nucleosome and then decompact the chromatin locally. You see? And the reverse process is exactly the same. There is also now in yellow another transcription factor that we will call a repressor. It's going to interact to recruit histone desacetyltransferase. So this enzyme is going to remove the acetyl group on the nucleosome, and the consequence will be compaction, and so the transcription cannot happen. You see what I mean? So the difference is a general transcription factor normally recognize the promoter and directly activate Pol2. And there are others that directly recognize DNA, but instead of activating directly Pol2, they recruit acetyltransferase in order to decompact chromatin locally. And because the chromatin is decompact locally, it is accessible to the Pol2 and the transcription will start. Okay? So basically, the consequence is the same. The consequence is transcription activation. But the way the transcription activation is achieved is not the same. In one way, the classical way is direct activation of POL2, and the non-direct way, the one that is on this slide, is activation through decompaction. You understand the differences?
Yes.
Tell me if you don't understand. We will see example in the coming days. I'm just checking the time.
So, can I ask?
Yes, of course.
Yes, because you say that the transcription factor interacts with DNA sequence to activate. So if in case of the indirect, where it binds to the chromosome or the chromatin to because you said that it's the need to interact with the chromatin first, then it rescues the acetylene. Yeah. So if the chromosome is so compact.
Okay. I see what you mean. If the chromosome is still compact, first it needs to be at least a little bit decompacted to interact. You need at least to have a little bit of decompaction. But I will show you what can happen, but the drawing is not really clear here because you can have the green transcription factor can interact, it can recognize the DNA on the nucleosome. The idea is this green transcription factor just need to recognize a part of the sequence, even if it's compacted, it can recognize because it's just a surface interaction, okay? And in the nucleosome, the DNA is around. So the transcription factor can recognize the sequence, but when he recognizes the sequence, he brings with him the histone acetyltransferase. And the consequence of the recruitment of the acetyltransferase will be to transfer acetyl groups. And then the transcription factor was in a compacted state, but after having the H80 complex, all the loci is going to be decompacted. Okay, I see what you, when I say compact chromatin, it's true that RNA POL2 cannot come because it's too big. But think about one thing is that the DNA is also at the surface of this loop around the nucleosome. So there are transcription factors that can still bind a little bit. And when they bind, if they bring this histone acetyltransferase, the consequence will be completely decompaction. And then other partners can bind properly after. Is it clear? Or?
Yeah, no? It's clear.
Okay. So, okay, we are, so I just wanted to talk to you about another type of post-translational modification, which is the methylation. So it's like acetylation, but it's a methyl group instead of acetyl. But we have methyl acetyl groups, that's just the reaction, and it's also analyzing amino acid. I just want to show you something. So here is just what we talked about before, but for the methylation of histone. So we have writers and readers. Okay, I'm not going to go in details in this. So the writers, so it means the enzyme or the complex that are going to methylate histone. So the code, for example, here is the methylation of histone 3 on the lysine at position 9. Here is the methylation of H3 on the lysine of position 27, and so on and so on. So forget about this slide, just to show you something that for the methylation, we know very well, like for acetylation, methylation is done through histone methyltransferase, exactly the same as histone acetyltransferase. There are different histone acetyltransferase. They can transfer an methyl group on different lysine, okay? And then there are readers, exactly as before. Readers means proteins that can recognize these specific marks. So, for example, HP1, I'm just going to talk to you about HP1. HP1 means heterochromatin protein 1. HP1 is going to interact with histone H3 only when the lysine at position 9 is methylated. Okay? So, let's say there is a writer which is called SURA. We're going to talk about that later on. This writer is able to transfer a methyl group to the lysine number 1 of histone H3. Okay, that's a mark. Then the reader, if this mark is present, this mark is methylated lysine at position 9 of histone H3. If HP1 is present, and if HP1 recognizes this kind of mark, HP1 is going to interact with H3. Okay? So this is the reader. Okay? I'm not going to talk to you about that. It's exactly the same drawing as before. When it's just to show you this, when the lysine at position 9 is methylated, HP1 recognize, it can read and interact with this. And the consequence of having HP1 linked to H3 is you have production of heterochromatin. Okay, it's going to come. Okay. I'm looking at the time. I think we need to stop. We almost finished the lecture. We will finish it tomorrow. Okay. Because I have a meeting in 10 minutes. We're going to take back the example of acetylation and methylation of the histone, and to see what I'm going to explain you is how these writers and these readers of the epigenetic marks, how they have been characterized. And we're going to see that from a historical point of view. Okay, so what we can do, we're going to finish now because I will be late after. Tomorrow we start at, I don't remember the time. We start at 4:30, okay, tomorrow. And what I would like you to do is look back the course of today, and if you have a question, and then we will finish the lecture with the suppression of variation tomorrow. Okay. So what I do now, I send the slides to Miss Hang, okay, and she will send you a mail how to download them. Okay. Is it okay, Rupa? Can you? Okay. So let's make the synthesis. I send you all the slides of the two lectures. For tomorrow, try to read back again the last part of the lecture of today. If you have questions, and then we will finish the lecture like that. Okay? Okay, thank you very much. Sorry, because if I had time, I could have finished, but I have another meeting with other people at USTH in 10 minutes. Okay, so write down your question and I will try to answer everything. Okay?
Okay.
Thank you.
So thank you and see you tomorrow at 4:30. We will probably finish before 6 p.m. Okay?
Okay.
Okay. Thank you very much and see you tomorrow. Okay?
Thank you.
See you tomorrow.
See you
Thank
tomorrow.
you.
Thank you very much.
Have a good day.
Have a good day also.